the United States launched a national campaign against cancer. The National Cancer Act carried the confidence of an era that had just placed human beings on the moon. Concentrate money, scientists, technology, and political will, and cancer might eventually surrender. Yet cancer still causes roughly 10 million deaths worldwide each year. Treatments are more precise, survival has improved, and several once deadly cancers are now manageable. But a universal cure remains out of reach. The reason is not that researchers failed or society ignored the problem. Cancer is unlike almost every enemy medicine has defeated. Cancer does not usually arrive from outside the body. It begins inside it. A bacterium carries foreign proteins. A virus introduces foreign genetic material, and the immune system can potentially identify that difference with infection. The mission is clear: destroy the invader while protecting the patient. Cancer destroys that clean boundary. A cancer cell is one of your own cells, built from your DNA, using your nutrients protected by your tissues. And using normal biological machinery that has become dangerously misregulated, the target is not a foreign organism. It is a distorted version of self that makes treatment an extraordinarily difficult targeting problem. Chemotherapy reveals the dilemma clearly. Many chemotherapy drugs attack cells that divide quickly. Cancer cells often divide quickly, but so do cells in hair follicles, the digestive lining, bone marrow, and parts of the immune system. The medicine can recognize a behavior, rapid growth, but it cannot always recognize identity with perfect accuracy. Consider the scale of ordinary human biology. The body contains tens of trillions of cells, and nearly every cell carries a copy of a genome containing about 3 billion DNA base pairs. Whenever a cell divides, that genetic information must be copied. The process is accurate, but not flawless. Small mutations appear across trillions of divisions during a lifetime. Most never matter because the body has layers of protection. DNA repair systems correct copying mistakes. Tumor suppressor genes slow inappropriate growth. Damaged cells can trigger programmed death, and immune cells patrol for abnormal behavior. Cancer develops when several protections fail in one cellular lineage, over years or decades. A cancer cell does not merely grow faster. It may ignore stop signals, resist death, attract a blood supply, hide from immune attack, invade nearby tissue, and eventually travel elsewhere. Modern cancer biology, therefore, treats a tumor as an evolving ecosystem, not a uniform mass. Cells inside one tumor can carry different mutations, compete for space and nutrients, and respond differently to treatment. When therapy kills the most vulnerable cells, a small resistant population may survive. Those survivors reproduce. And the returning tumor is now enriched with traits that helped it endure the first attack. Treatment imposes selection pressure, allowing the best adapted cancer cells to dominate. That moving target is one reason a drug can work powerfully at first, then lose effectiveness later. The phrase curing cancer hides another problem. Cancer is not one disease. It is a category containing more than 200 diseases. A leukemia arising in blood-forming tissue, a melanoma beginning in pigment-producing cells, a glioblastoma growing in the brain. And a breast tumor driven by a particular mutation may share uncontrolled growth, but they do not share one identical biology. Even to patients with the same diagnosis and stage can have different mutations, immune environments, metabolic needs, and vulnerabilities. A therapy that helps one patient may do little for another, because the name of the cancer does not reveal every molecular detail. Personalized medicine, therefore, matters. Researchers can sequence a tumor, identify mutations that appear actionable, and select therapies designed for those targets. That approach has produced success, but the tumor may change during treatment. A biopsy offers a snapshot, while cancer keeps evolving. The immune system creates another layer of hope and difficulty. The principle: immune cells should recognize abnormal cells and remove them before a tumor becomes established. Much of the time, it does. The cancers that become clinically visible are often the ones that learned to escape. Some cancer cells display signals that tell immune cells to stand down. Some reshape the tissue around them into an immunosuppressive environment. Some recruit normal immune cells and redirect them to protect the tumor. Immunotherapy attempts to reverse that deception. Checkpoint inhibitors can release molecular brakes that prevent immune cells from attacking. And in some patients with melanoma, lung cancer, and other cancers, the results have been extraordinary. People expected to survive months have remained alive for years. Yet these medicines help only a portion of patients, and removing immune restraints can cause the immune system to damage healthy organs. Those restraints evolved to prevent autoimmunity. Cancer exploits them for protection, but releasing them creates a dangerous trade-off. Attack too weakly, and the tumor survives. Attack too broadly, and normal tissue suffers. Metastasis makes the challenge worse. The original tumor can often be removed by surgery or treated with focused radiation. The greatest danger begins when cells escape, enter blood or lymphatic vessels, survive circulation, settle in distant organs, and establish new colonies. A patient may face tumors in several organs, each in a different environment, and each evolving resistance independently. Metastatic cells must complete a remarkable sequence of biological tasks: detach, travel, evade immunity, penetrate unfamiliar tissue, adapt, and restart growth. Each step suggests a target, but the same mechanisms also support healing, immune movement, and tissue maintenance. Blocking cancer without blocking essential biology remains the central difficulty. Dormancy makes the picture more unsettling. Some cancer cells stop dividing and enter a quiet state. They may become difficult to detect on scans and difficult to kill with drugs aimed at rapidly dividing cells. Years later, under conditions that are still not fully understood, they can become active again. Doctors therefore speak of remission and 5-year survival rather than promise cancer can never return. The laboratory creates another obstacle. A treatment may eliminate tumors in mice, produce exciting headlines, and then fail in human trials. That pattern does not prove fraud or incompetence. It reflects the gap between an experimental model and a human life. Laboratory tumors may be genetically simpler, grown under controlled conditions, or studied in animals with altered immune systems. Human tumors arise over decades inside people with different genes, ages, exposures, medications, and immune histories. Researchers are improving models through patient-derived organoids, more realistic animal systems, and computational simulations. But no model can reproduce every feature of a living patient. Early detection may therefore save more lives than any single dramatic treatment. The earlier a cancer is found, the less time it has had to spread and diversify. Stage 1 breast cancer has a 5-year survival rate above 90%, while Stage 4 disease has a rate below 30%. Biology matters, but timing can transform the outcome. Liquid biopsy is promising. Tumors release small traces of genetic material into blood, and researchers are developing tests designed to detect those signals before symptoms appear. The difficulty is finding tiny signals without causing false positives, fear, and unnecessary procedures. Large trials must show not only that a test detects cancer, but that using it actually reduces deaths. The economics of research also shape progress. Developing a cancer drug can cost billions, take years, and fail late in trials. Companies therefore favor products capable of earning back that investment. Treatment is often easier to finance than prevention, because medicine for identifiable patients has a measurable market, while a cancer that never develops leaves no visible customer and no obvious proof of prevention. Public research fills part of this gap, but grant deadlines and publication incentives can reward safer, incremental projects over risky work that may require many years to mature. Progress is not imaginary. Childhood leukemia, once almost universally fatal, is now curable in roughly 90% of cases. Hodgkin lymphoma is highly treatable. Several early breast cancers have excellent survival rates, and engineered CAR T cells have produced complete remissions in some blood cancer patients after other therapies failed. The pattern is clear. Success comes fastest when a cancer has a clear driver, is accessible to drugs or immune cells, or is biologically less diverse. The hardest cancers, including glioblastoma, pancreatic cancer, and many metastatic solid tumors, are often genetically complex, difficult to reach, and skilled at suppressing immunity. The next era will combine tools rather than depend on one miracle. Artificial intelligence can compare enormous tumor datasets, connect mutation patterns with treatment responses, and help identify targets that human researchers might miss. Messenger. RNA platforms may support personalized vaccines that train the immune system to recognize proteins unique to an individual tumor. Better sequencing, better imaging, earlier detection, improved immunotherapy, and more accurate models are beginning to converge. The conclusion is not that cancer can never be controlled. It is that there will probably be no single day when one universal cure defeats every form of it. Cancer is built from normal biology, changes under pressure, hides within the body, and appears as hundreds of distinct diseases. Progress will come cancer by cancer, mutation by mutation, and patient by patient. Some battles are won, others are being won, and many remain painfully difficult. The campaign begun in 1971 was never one war against one enemy. It was hundreds of overlapping wars against diseases that evolve while we fight them. The scientists confronting those diseases are not standing still. They are learning how each cancer survives, where it is vulnerable, and how to intervene with less damage to the person carrying it. In medicine, that accumulation of knowledge may look slower than victory, but over time, it is how victory is built.